365. Myself Srija. So today in this session we are going to learn about physiology of immune system. So physiology of immune system explains how immune cells work, how tissues and chemical molecules work together to defend the body against infections, toxins, cancer cells and damaged or abnormally body cells. So think of the immune system as the body's security department with workers, sensors, alarms and memory. So overview of immune physiology. So immune function, it can be understood through three major steps. So this is the first and most crucial step, recognition. So what is recognition? What happens here? The immune system identifies bacteria, viruses, fungi, parasites, toxins, cancer cells and damaged or dead cells. So immune cells, they use receptors to detect the molecules called antigens on the surface of the pathogens. So what are the key cells in the recognition? Macrophages, dendritic cells, neutrophils, natural killer cells, B cells and T cells. So these cells, they act as the body security cameras, scanning for the danger. So recognition is like a sensor detecting a burglar entering a house. So the second important function is response, attacking the threat. So once a threat is recognized, the immune system activates a coordinated defense response. So what happens here? First, the immune cells become active, inflammation occurs, antibodies are produced, killer cells destroy the pathogens or infected cells. So the complement proteins help to punch the holes in the bacterial membranes. So what are the types of responses you have? The first, innate response and adaptive response. So the innate response is an immediate response. It is very fast, like minutes to hours. It is non-specific. It includes fever, inflammation, phagocytosis. It involves macrophages, neutrophils as well as NK cells. And the second response is adaptive response, which is a specific one. So it is very slower. It lasts for days. It is highly specific. So it involves the both B and T lymphocytes. It produces antibodies and memory cells. So response is like the security team catching the burglar and stopping the threat. So the third important function is memory. So after the threat is destroyed, the immune system stores information about the pathogen. So why memory is important? Because it helps the immune system to respond faster and stronger the next time. So it prevents the reinfection or it reduces the severity. So what are the memory cells involved over here? Memory B cells and memory T cells. So memory B cells, they remember the antigen and produces the antibodies faster. Whereas memory T cells, they respond quickly to previously seen infections. So examples like chickenpox infection, which gives a lifelong immunity, measles vaccine, long-term memory, booster doses, strengthen the memory. So memory is like saving the burglar's face in the data phase. So next time security reacts instantly. So if we put it all together, recognition, immune system sees the intruder, nothing but a pathogen. And response, immune system attacks and removes the pathogen. Whereas memory, the immune system saves the intruder's identity for future protection. So these three steps, they create a complete, efficient immune defense that protects us every single day. So physiology of innate immunity. The second function is, so innate immunity is the body's first immediate and non-specific defense system. It protects you from infections even if your body has never seen that microbe before. So it mainly works through physical barriers and chemical barriers as we have discussed earlier. So again we'll discuss about this physical and chemical barriers. So skin. So what skin does do? Skin is the largest and most important protective organ of the immune system. So how skin protects you? Through keratin. It produces keratin. It acts like a mechanical barrier. So the outermost skin layer is filled with keratin, a tough protein. This makes the skin waterproof and difficult for microbes to penetrate. And also it has the sebum, which is the oily secretion from sebaceous glands. So this contains the fatty acids which have low pH value. So sebum, it creates an acidic environment where bacteria cannot grow easily. And sweat. So sweat contains lysozyme and enzyme. It breaks bacterial cell walls. So this is how skin protects you. So next is mucous membranes. So this line the respiratory, digestive, urinary and reproductive tracts. This is the areas where microbes can enter. So their protective mechanisms is the mucus secretion. So sticky mucus, it traps dust, bacteria and viruses like a fly paper. And cilia, cilia action, it is present in the respiratory tract. So these are the tiny hair-like structures which move rhythmically. So they push the trapped microbes upward where they can be coughed out or swallowed. So next is stomach acid HCl. So the stomach is extremely acidic. So pH value ranges from 1 to 3. So how does it protect? It kills mostly swallowed pathogens such as bacteria in the food or water. So it destroys the parasites and it prevents the intestinal infections. So for example, if someone eats contaminated food, stomach acid is the first weapon against this infection. And saliva and tears. So these fluids, they constantly wash the surfaces and contain the natural antimicrobial substances. So what is the key protective component present in saliva and tear? Lysozyme. So this is the same enzyme which is found in the sweat. So it breaks down the bacterial cell walls, the bacteria burst and die. So it also has the extra protective actions, the flushing action. So tears wash away microbes from the eyes, whereas saliva, it clears the mouth and prevents the bacterial buildup. So why these barriers are important? Because they work immediately. There is no need for previous exposure. So they protect against a wide range of microbes which are non-specific, and they also prevent many infections before the immune system even realizes there is a threat. So next is phagocytosis physiology. So what is phagocytosis? It's a process where certain immune cells eat and destroy the microbes. So the main phagocytes include neutrophils, which are the first responders, short-lived, but they are the fast killers. Macrophages, they are the long-lived and they are powerful big eaters. Dendritic cells, these are also phagocytic, but they are mainly important for antigen presentation. So what are the steps of the phagocytosis? Chemotaxis, nothing but finding the enemy. So phagocytic cells, they move towards the infection site, injury or microbes releases the chemical signals. So this pull the phagocytes towards the damaged area, like a firefighter following smoke to find the fire. The second step is adherence, catching the enemy. The phagocyte attaches to the microbe surface. So this attachment, it is strengthened by opsonins, the antibodies or complement proteins which coat the microbe. And the third step is engulfment, swallowing the enemy. So the cell membrane extends around the microbe, which is forming a bubble-like sac called as phagosome. So this is like wrapping the microbe inside a sealed bag. And the next is fusion, merging with the killing chamber. So the phagosome forces with the lysosome, which is a cell organelle, which is full of digestive enzymes. So this forms a phagolysosome. So the lysozyme is like a toxic chemical chamber inside the cell. And the sixth step is digestion. So killing and breaking down the microbe. Inside the phagolysosome, there are enzymes like proteases and lipases, and there are also like peptides, antimicrobial peptides. So what does they do? They kill and break down the microbes. So they are the reactive oxygen species which destroys the pathogens. So this is a step where the microbe is chemically burned and digested. And next step is exocytosis, throwing out the trash. So any leftover waste is expelled out of the cell. And here is a diagram on the right side which shows you the journey of a pathogen inside a phagocyte. So the first step is recognition or binding. So receptors on the phagocyte recognize the patterns on the microbes. So microbes bind to receptors, then it gives signal for engulfment begins. So the second step is target internalization. So the membrane extends around the pathogen to form a phagosome. So the third one is phagosome maturation. So the phagosome changes over time, early phagosome and late phagosome. Later on it fuses with the lysozyme and it forms the phagolysozyme. So the fourth is target lysis or digestion or killing. So inside the phagolysozyme, there are ROAs, DNAs, proteases which destroy the microbe. So you can see the microbe breaking part. So the last one is fifth one, the early phagosome, the waste removal. So digested remnants are released outside by exocytosis. So this is how the pathogen inside a phagocyte reacts. So next is inflammation physiology. So inflammation, it is a protective response which is triggered whenever tissues are injured or infected. So what is the main goal of this inflammation? To remove the cause of the injury, to remove the dead cells and to begin the healing. So there are steps of inflammation. So the first step is tissue injury or infection. So damage to the skin or tissue or entry of the microbes signals the immune system. So second is the release of inflammatory mediators. So these are the cells at the injury site. They releases the histamine from mast cells. So this histamine, it causes the vasodilation and it increases the blood flow. Whereas prostaglandins, it increases the pain, fever and inflammation. And bradykinin, it increases the pain and swelling. So these chemicals together, they create the classic signs of inflammation. So there are four cardinal signs of inflammation: redness, heat, swelling and pain. Redness is due to increased blood flow. Heat is due to warm blood coming into the area. Swelling is due to fluid leaks out of the blood vessels. And pain occurs due to chemicals stimulate the nerve endings. So why inflammation is beneficial? Because it brings more immune cells to the infected area. It makes blood vessels leaky, so phagocytes can escape into the tissues. So generally people often think inflammation is harmful. So you have to emphasize the acute inflammation is protective. Always remember, acute inflammation is protective. So next, complement system physiology. So what is complement system? It is a powerful part of innate immunity which is made up of more than 30 plasma proteins circulating in the blood in an inactive form. So when activated, these proteins work together like a cascade of falling dominoes to destroy the microbes, enhance the inflammation, and it supports the phagocytosis. So why is the complement system important? Because it puts a label on the microbes so phagocytes can easily catch them. So it creates the chemical signals that call more immune cells into the battle. It can also even punch holes in the microbes to kill them directly. So what are the major functions of the complement system? So the first one is opsonization, tagging the enemy. So C3b, which coats the pathogen surface. Phagocytes, it have receptors for C3b. So this makes the phagocytosis much faster and easier. Second is cell lysis, nothing but MAC formation, the membrane attack complex formation, drilling holes in the microbes. So complement proteins like C5b, C6, C7, C8 and C9, they form the membrane attack complex. So MAC, it inserts into the microbe cell membrane and forms a pore. So water rushes in through the microbe burst and dies. So this is especially important for killing gram-negative bacteria. The third one is trigger inflammation. It is done by C3A and C5A complement proteins. So these are also called as anaphylactoxins. And they can attract the neutrophils and macrophages. So they can increase the blood vessel permeability and also intensify the inflammation. So C5A is the strongest chemoattractant in the complement system. And the next function is clearing immune complexes. So complement helps remove antigen-antibody complexes from the blood. So it prevents them from depositing in the tissues and reducing the autoimmune damage. So what are the activation pathways? So how the complement system is switched on? So there are three different pathways, but all lead to the activation of C3 to C5, then to membrane attack complex. So the first pathway is classical pathway, which is activated by antigen-antibody complexes. So usually by IgG or IgM. So this is used when the adaptive immune system is already responding. And the second pathway is lectin pathway. So this is activated when mannose binding lectin, MBL, it binds to sugars on microbial surfaces. So it does not require antibodies which are useful in early infection. So next is alternative pathway. So this is activated spontaneously when C3 is hydrolyzed in the plasma. So alternative pathway, it is powerful because it does not need antibodies or lectins. So it is often the first pathway to respond. So what all pathways leads to the common final sequence? So all three pathways, they converge at C3 convertase formation. So this splits C3 into C3A and C3B, nothing but opsonization. So it also converts C5A, C5 convertase. So it splits C5 into C5A and CB5 triggers the MAC formation. So here there is a diagram showing you the Three entry points: classical pathway, which is triggered by antibody binding; lectin pathway, which is triggered by MBL; so C5b, it triggers MSC formation, a membrane attack complex. So here is a diagram which shows you the classical pathway, lectin pathway, and alternative pathway. So lectin pathway, it is triggered by MBL binding, and alternative pathway, it is triggered by the spontaneous activation. So all three arrows, they lead towards C3. So C3 activation. So C3, it splits into C3a, C3b. So C3a promotes the inflammation, whereas C3b, it attaches to the microbes. Opsonization plus it helps from C5 convertase. So you see C3a going upward towards the recruit neutrophils, and C3b is shown coating the microbes. And then C5 activation. So C5 splits into C5a and C5b. So C5a, it is a very strong inflammatory mediator and chemoattractant. And C5b, it starts MAC formation. So the diagram shows C5a going upward or recruit neutrophils and monocytes. So then MAC assembly. So right side of the diagram, C5b, it interacts with C6, C7, C8 and C9. It forms a cylindrical channel in the microbes membrane which causes a cell lysis. So diagram shows a purple pore inserted into target cell membrane. So this is about the complement system physiology. So then NK cell physiology. So what are NK cells? Natural killer cells. These are the cells which are type of innate immune cell that act as a body's rapid response team. So they do not require prior exposure to an antigen, unlike T cells. So they are especially important in detecting the abnormal cells early. So NK cells kill the virus infected cells. Virus often hide the inside host cells. So NK cells, they patrol the tissues and destroy the cells, showing the signs of viral infection. And also tumor cells. So cells that start becoming cancerous, they show the abnormal or reduced self markers. So NK cells, they recognize these abnormalities and they eliminate such cells early, preventing tumor formation. So now what is the mechanism of NK cell killing? It releases the perforin which forms pores or holes in the target cell's membrane. So perforin, it performs as a drill that creates the entry points. And then granzymes. So granzymes, these are the enzymes that enter through the perforin made pores. So they trigger the apoptosis, a clean programmed cell death. So apoptosis, it prevents inflammation and it stops the infection spread. So next, coming to physiology of adaptive immunity. So adaptive immunity, it is a part of the immune system that is very specific and highly regulated. It forms the memory and it has a slower initial response, but it becomes faster upon secondary exposure. So there are two major types of adaptive immunity, humoral and cell mediated, as we have discussed in the earlier session. So what does happen in humoral immunity? What do B cells need to be get activated? Antigen binding. So B cells, they have surface immunoglobulins which are B cell receptors. So when an antigen binds, the B cell receives its first activation signal. So then helper T cell signals, cytokines. So these helper T cells recognize the same antigen which are presented by B cells. So they release cytokines such as IL-4, IL-5 and IL-6. So these cytokines, they give the second activation signal. So without helper T cell signals, B cells produces a weak response. With higher T cell, they help to the B cells produces a strong, long-lasting immune response. So what happens after activation? B cells differentiate into plasma cells like antibody factors. It produces large amounts of IgM, IgG, IgA and IgE. And also it differentiates into memory B cells. So they live for years. They respond quickly upon re-exposure. So next is B cell differentiation. So when a B cell becomes activated, it divides and transforms into two important types. The one is plasma cells, which are antibody factors. The other is memory B cells, which are long-term protection. So these plasma cells, these are the cells which produce large amounts of antibodies. So their job is to neutralize and eliminate the pathogens circulating blood, lymph and mucosal areas. whereas memory B cells, those remain in the body for years. So they respond rapidly and strongly when the same pathogen appear again. And antibody physiology. So antibodies, they perform several key protective functions. So they do not kill pathogens directly. They guide other immune mechanisms. So what are the major antibody classes and functions? IgA, this is most abundant one. It crosses placenta and it has long-term immunity. IgM, it is the first antibody produced and it is very strong at agglutination. And IgA, mucosal immunity. It secretes tears, saliva and gut. IgE, allergic responses. So defense against the parasites. And IgD, which is present on the B cell surface for activation. So how antibodies neutralize pathogens? Antibodies help the body in several ways, like neutralization. Antibodies bind to toxins or viral proteins. This blocks attachment of viruses or bacteria to these host cells and they prevent the infection entry. Second is absonization, like tagging for eating. So antibodies coat pathogens, makes them easier for macrophages and neutrophils to engulf. Agglutination, clumping. So antibodies bind multiple pathogens at once. So clumps them together, which is easier for phagocytes to clear. And complement activation. Antibody coated microbes activate complement proteins. So which leads to formation of macropores, cell lysis and enhanced inflammation. So next is cell-mediated immunity, the T cell physiology. So this part of immunity deals with the infected cells, especially viral or intracellular infections. So helper T cells. These are the command cells of the immune system. They do not kill directly, but they coordinate the entire immune response. So what are the functions of the helper T cells? They secrete the cytokines. So they are the chemical messengers that guide other immune cells. So they activate the B cells to form plasma and memory B cells. They activate macrophages to make them better at killing. They activate cytotoxic T cells which are enable killing of the infected cells. And this helper T cells, they also regulate the entire immune response. So here is a diagram which shows you the humoral response and cellular response. So cytotoxic T cells. So these T cells, they are also called as killer T cells because they directly destroy the infected or abnormal cells. So what are the functions? They kill the virus infected cells. They kill the cancer cells. So they can recognize the antigens present on the MHC class 1 molecules. So what is the mechanism of the killing? CD8 plus T cells, they release both perforin and granzymes. So this method, they prevents the inflammation and ensures the clean removal of infected cells. And coming to regulatory T cells, which is also called as Tregs. So they act as the police of the immune system. So what are the functions? They prevent overreaction of the immune system. They maintain tolerance. Like they prevent the body from attacking its own tissues. They release IL-10 and TGF beta to control the excessive inflammation. So they prevent the autoimmune diseases such as type 1 diabetes, rheumatoid arthritis and lupus. So without Tregs, the immune system becomes uncontrolled, which results in autoimmune diseases. So next is antigen processing and presentation physiology. So the immune system, it cannot recognize an antigen directly. So it must be processed and presented on a special molecule called MHC, the major histocompatibility complex. So this is done by the antigen presenting cells, which are called as dendritic cells, macrophages and B cells. So there are two major pathways of antigen presentation. The first one is MHC class 1 pathway, which is an endogenous pathway, and the second pathway is MHC class 2 exogenous pathway. So the left side, there is a diagram. So the class 1 endogenous pathway. So there are endogenous proteins. These are from viruses or abnormal cells. So these are broken down by the proteasome. So proteasome is a protein shedder. So the resulting peptides move into the endoplasmic reticulum through a tap transporter. So inside the endoplasmic reticulum, the peptides bind to MHC class 1 molecules. So the MHC1 peptide complex is transported via the Golgi apparatus. So it is expressed upon the cell surface. So then CD8 plus cytotoxic T cells, they recognize the antigen MHC1 complex. So they kill that cell. So MHC1 is present on all the nucleated cells. They present the intracellular antigens. So it always activates the CD8 plus T cells. So coming to exogenous pathway, the class 2, MHC class 2 pathway. This pathway, it presents extracellular antigens such as bacteria, parasites, toxins. So anything which is taken in from outside the cell. So the first step is APC, like dendritic cell, it ingests the pathogen by phagocytosis and endocytosis. So the antigen is broken into small pieces inside the endosomal compartment or lysosomal compartment. So meanwhile, the MHC class 2 molecules, they are made inside the endoplasmic reticulum. So MHC class 2, it is protected by invariant chain. It is a clip. So peptides don't bind in the endoplasmic reticulum. So in the endosomal compartment, what happens? Invariant chain is removed by HLA-DM. So antigen peptides, they bind to the MHC class 2 molecule. So this MHC2 peptide complex, it is transported then to the cell surface. And at last, helper T cells CD4+, they recognize the antigen MHC2 complex. So then CD4+ tells T cells, they activate B cells for antibody production. They activate macrophages to enhance the killing, and they activate the CD8 T cells indirectly. So they also coordinate the entire immune response. So next is cytokines, the communication system. So cytokines, these are like the mobile phones or signal messages which are used by the immune cells to communicate and coordinate actions. So they are small protein messengers produced by T cells, B cells, macrophages, dendritic cells, endothelial cells, fibroblasts, and mast cells. So there are major types of cytokines. So interleukins, IL-1 to IL-38. So what is interleukin? What do you mean by this? It is present between leukocytes, like WBC communication. So IL-1, it causes fever. IL-2, it causes T cell growth. So IL-4, B cell activation, and IgE class switching. Like every interleukin has its specific function to perform. And there are major types of interferons. So interferon alpha, beta, gamma. So these are very primarily antiviral. So they block the viral replication, they activate the NK cells. Interferon gamma, they activate the macrophage, it promotes the Th1 responses. And TNF alpha, the tumor necrosis factor alpha. So this is a strong pro-inflammatory cytokine. These are produced by the macrophages. So what does it cause? It causes fever, inflammation, and recruitment of the immune cell. So what is the function of the cytokine? It induces the inflammation. It increases the blood flow. It causes the redness, heat, and swelling. It also recruits the immune cells to the site of the infection. And second function is it activates and attracts the immune cells. Like it acts as a chemical signals. For example, IL-8, it attracts the neutrophils. CCL2, it attracts the monocytes. So cytokines, it also promotes T cell and B cell activation. So it controls fever. Like fever improves the immune efficiency. So here is a diagram which involve cytokines. So lymphocytes, macrophages, granulocytes, fibroblasts, and endothelial cells and mast cells. So these are the different immune cells which sends and receives the cytokines like a communication network. So the next function is immunological memory. So immunological memory is one of the most important features of adaptive immunity. It is what allows your body to respond quickly when it sees the same pathogen again. So what is immunological memory? After you get an infection or receive a vaccine, so your immune system does not forget, right? So it stores the information about the pathogen so that if it enters again, the body reacts much faster. So what is a memory cell physiology? Like how memory cells work. So after infection, some B cells and T cells become memory cells. So when your body fights a germ, B cells and T cells gets activated. So after the infection is cleared, most of the cells die. But some survive as long-living memory cells. So these memory cells, they persist for years or decades. So they live in lymph nodes, bone marrow, and spleen. They can survive about 10 to 20 years, sometimes for life even. And secondary immune response is faster and stronger. The primary response, it is very slow and weaker, whereas secondary response, it is very fast within hours and very strong. It has very high antibodies. So this is why you usually do not get severely sick with the same infection again. And the next physiology is basis of vaccination. So vaccines introduce a safe version of a pathogen, whether it is a weakened or dead. So your immune system, it forms the memory cells. So the next time the real pathogen attacks, your body immediately fights it. So this is how the immunological memory works. And then physiology of fever, pyrexia. So what is fever? Fever is a controlled, regulated rise in body temperature. It is not random. The body intentionally increases temperature to fight the infection. So what is the mechanism of fever? Here is a step-by-step process. The first step is infection occurs. So bacteria or viruses enter the body. So then the macrophages detect the pathogen. Macrophages is nothing but they are the soldiers of immunity. And then macrophages releases cytokines. So the cytokines, these are the fever producing chemicals. So these are called as endogenous pyrogens, which are internal fever causing substances. So the cytokines, they act upon the hypothalamus. So hypothalamus, then it increases the prostaglandin E2. So the prostaglandin E2, it changes the body's temperature set point to a higher level. And then what happens? It raises the body temperature to reach the new set point. So the body performs shivering to generate the heat and vasoconstriction to reduce blood flow to the skin. So this helps to raise the body temperature. Now what is the purpose of fever actually? So fever is actually protective. It is not harmful unless it is very high. So it slows the pathogen growth. So most of the bacteria and viruses, they grow poorly at higher temperatures. And fever, it also enhances the immunity. It improves the white blood cell activity. It enhances T cell function. It speeds up the chemical reactions of immune cells and it promotes the faster healing. So this is the purpose of the fever. And this is for today's session. I hope this session has helped you. So in the next session, you will be able to learn the ICD-10 coding guidelines for chapter 1 and chapter 2 within the ICD-10. Thank you.